Hey, what's going on, guys? My name is Matthew Joes, and today we're bringing you the physiology behind the cardiac output and how it's regulated and controlled. So this is a video in the series of the cardiovascular physiology. So cardiac output is basically the volume of blood pumped by each ventricle contraction per minute. So the main thing that you want to be aware of is the fact that it's per minute, right? And cardiac output is calculated by stroke volume, SV, times heart rate, and I will go into detail of the different definitions of what stroke volume is and heart rate. But just for your understanding now, stroke volume is the volume of blood pumped out by each ventricle per beat. Okay, it's not per minute, per beat. And cardiac output is per minute, right? And heart rate is how much times the heart beats in 1 minute. So to get cardiac output, all you do is the volume of blood pumped out per beat times by the number of beats in a minute, which is heart rate. So an example is shown. Someone who has a stroke volume of 70 mil per beat and an heart rate of 72 beats per minute would have a cardiac output around 5,040 milliliters. So if you would convert that into liters, it's roughly around 5 liters. This is the normal resting rate. So at rest, the cardiac output is roughly in a normal healthy adult of around 5 liters per minute. And it can be during stress such as exercise, it can be increased as much as 4 to 5 times, so pushing out around 20 to 25 liters per minute. And really, really fit people such as athletes and those who train pretty much 6 to 7 times a week can increase the cardiac output by much as 7 times that of the resting rate, which means they can push about 35 liters per minute. And this is known as the cardiac reserve. So the heart rate, as I mentioned, the cardiac reserve is the maximum amount of the cardiac reserve is actually the difference between the maximum that someone can push, the maximum amount of blood that they can push per minute, take away the maximum blood they can push at rest. So in this case, in the case of an athlete, it will be 35 liters per minute, take away 5 liters per minute. So the cardiac reserve for them would be 30 liters per minute. Now, heart rate is basically the number of ventricular contractions per minute, and stroke volume is the amount of blood pumped out of the ventricle with each contraction. Okay. Now, regulation of stroke volume. So stroke volume is basically calculated via end diastolic volume, take away end systolic volume. And end diastolic volume and end systolic volume, I've explained it in a very great detail in the video which shows the cardiac cycle. But just to give you a rough idea again, end diastolic volume is the amount of blood collected in a ventricle during diastole. So it's the maximum amount of blood that the ventricles contain within itself in the very late stages of diastole, which means in the very late stages of ventricular relaxation, just before it's about to contract. And ESV is the amount of blood remaining in the ventricle post-contraction. So systolic volume, stroke volume basically is the amount that's pushed out. So all you do is to find the difference between the EDV and the SCV. Now, you know the variables that determine cardiac output. It's the heart rate and stroke volume. And since they have a direct relationship, if heart rate was to increase, you would expect cardiac output to increase. And similarly, stroke volume, if that was to increase, cardiac output would also increase with it. Now, variables that determine CO. So force of the heart muscle contraction. The contractility is a variable that determines how much determines the cardiac output, and this contractility comes down to the force of the heart muscle contraction. So if your heart was to contract with a greater force, you're inevitably pushing out more blood at this in one go compared to if you were at resting. Or so, for example, if you're pushing with 50 newtons, you're pushing more blood compared to if you were pushing with a force of 20 newtons. Now, extrinsic control factors from outside of the heart include things like which determine the cardiac output includes neural input as well as hormonal input such as drugs, neurotransmitters, and which we will go into a bit more detail in the latter stages of this video. Intrinsic, so there's an intrinsic control and an extrinsic control. The intrinsic control includes factors from within the heart, and this is stuff like the Starling's law of heart, which includes preload and afterload, whereas extrinsic control is via the autonomic nervous system, which is the neural input as well as the hormonal input. Now, we've established so far that the cardiac output is, depending on both heart rate as well as stroke volume, and whatever changes which is caused to the heart rate, it would have a direct impact on the cardiac output. So if the heart rate was to increase, the cardiac output would also substantially increase. So so in this slide, it shows you what are the different changes that control, the heart rate and inevitably the cardiac output. So you have the autonomic control, which is split into sympathetic and parasympathetic. So the heart rate is influenced by three types of factors. You have the sympathetic, which increases heart rate, parasympathetic, which decreases the heart rate, and you also have hormonal control. Now, most of the nerve fibers of the ANS, i.e. the sympathetic and parasympathetic, directly affect the SA node as well as the AV node, and the ventricular myocardium itself. So, the muscle that surrounds the apex of the heart. The ANS, so the parasympathetic and sympathetic, control both heart rate as well as stroke volume, stroke volume is basically contractility. Now, on the screen now, you have an image of the parasympathetic and sympathetic cardiac nerve. So this is just to give you an idea. Don't expect you to know it off by heart. And this is an image taken from the 2002 Pearson Education publications. So on top, you can see the medulla oblongata located, in the brain stem. The vagus nerve, which is cranial nerve 10, which is a parasympathetic nerve, has a direct nerve going straight down into the SA node as well as the AV node. So at times where your heart needs to relax and it needs to slow down the number of heartbeat, or it needs to decrease the cardiac output, the parasympathetic nerve, so the vagus nerves, is the one that signals the SA node and the AV node. Now, in times of in a flight and fight response, so i.e. when you've just had some sort of shock or your heart is beating really fast, this is because the sympathetic cardiac nerve, which is the sympathetic nerve, you can see it travels down the medulla oblongata into the spinal cord and through the peripheral ganglion and into the SA node as well as the AV node, and it can also supply the ventricular myocardium, so causing so in this case the sympathetic one will signal the SA node to send out more electrical signals and the AVN to send out more electrical signals, and the ventricular myocardium to contract with a higher force, hence all increase in the heart rate and stroke volume. So this is basically the same thing as what I've just mentioned. So the sympathetic nervous system stimulation is activated by stress, anxiety, or excitement or exercise. Parasympathetic is basically mediated by acetylcholine, the neurotransmitter, and opposes the SNS. So it's usually during digest and rest, so it decreases the heart rate. Usually the PNS dominates the autonomic stimulation, slowing the heart rate and causing vagal tone, because at most, in predominant amount of time that you spend your awake or you live during a day, you are not excessively under stress, or you're not excessively under anxiety or doing some sort of stressful exercise. You're most of the time in the world you're relaxed, so therefore it's PNS that dominates more than the SNS. So if the vagus nerve was cut, the heart would lose its tone, thus increasing the heart rate by around 25 beats per minute, which can be detrimental because it would then increase the heart rate. Now, factors affecting cardiac output include, we've already mentioned, heart rate and stroke volume. So the parasympathetic nervous system activation causes decrease in heart rate as well as decrease in stroke volume. And the parasympathetic nerve that you need to be aware of is the cranial nerve 10, vagus nerve, and it emerges from the medulla oblongata and primarily innervates the SA node and AV node. There will be another video subsequently later on in the cardiovascular physiology with the nervous control of the heart and how they're controlled in a completely separate video. So if you were to watch this video, you should go down after this one and watch that, just to reimburse and anchor the knowledge that you gain from both of the videos, so it really makes sense. Now, and the neurotransmitter involved in the parasympathetic nervous system is the acetylcholine. Now, sympathetic obviously does completely opposing, so it increases heart rate, stroke volume, and yeah, it's the cardiac nerve, like I said, from the sympathetic trunk of the ganglion, and it has innervations to the SA node, AV node, ventricular myocardium as well. And the neurotransmitter in this case is norepinephrine. Right, the other one was acetylcholine, but this time is norepinephrine, or you can, in terms of UK or a European student, you would refer them to as noradrenaline. So this is the one that really kicks in to increase your heart rate. So again, the same image that we've looked at before. So increased sympathetic activity, what does it do? So it increases the action potential frequency, as I've mentioned, shortens the time it takes for action potential to travel through the ventricles. So basically, all in all, reduces the delay and increases the heart rate, and therefore increases the cardiac output. Yeah, so the increased parasympathetic activity means decreased depolarization, decreases action potential frequency, and then the action potential is transmitted slowly. So therefore, it has a decrease in heart rate, therefore decrease in cardiac output. Now, hormonal control of the heart rate. So we've established there's a neural control, which these are all extrinsic controls of the cardiac output. So the hormonal control of heart rate include things like epinephrine. So this is noradrenaline. This is a catecholamine, and it's secreted by the adrenal medulla, and it's usually in response to the sympathetic nervous stimulation. So if you remember back, sympathetic nervous stimulation, the neurotransmitter involved is noradrenaline or norepinephrine, and that then stimulates the adrenal medulla to release epinephrine, which is adrenaline, which is a hormone, whereas noradrenaline is a neurotransmitter. So there's a fine difference between both. Now this travels through the bloodstream to the heart and exerts minute by minute control. So it increases the frequency of action potentials generated by the SA node, thus heart rate, and the heart rate is increased. Now, other hormones that increase heart rate include things like thyroxine, and you have different types of thyroxine. You have T3, T4, T5, but for your awareness so far, you should just know it as thyroxine, which is produced by the thyroid, located in your pharynx. This causes proliferation of androgenic receptors, the binding sites of catecholamine, resulting in heart rate. So basically, thyroxine's job is to increase the androgenetic receptors. Androgenic receptors are the receptors present on the surface of the heart, and these receptors are the ones to which the catecholamines like adrenaline or epinephrine bind to. So increasing the receptors means you have there's more space for the adrenaline to bind to. So therefore, inevitably, it increases heart rate, stroke volume, and cardiac output. And yeah, so inadequate thyroid function. So people who suffer from inflamed thyroids or inadequately working thyroid, they can potentially decrease heart rate and then decrease stroke volume, which can have a detrimental effect on their cardiac output. Now, so far we've gone through, so this is just the complete overview of what we've gone through so far. The three influences that control the heart rate include sympathetic, parasympathetic, and hormonal. So the parasympathetic dominates the heart at rest, but and it's controlled by the neurotransmitter acetylcholine, and the effect of it is it results in decreasing heart rate, so decreasing cardiac output. Now, the decreased parasympathetic input means that there's an increased sympathetic input, which is done by the neurotransmitter norepinephrine or noradrenaline. This then increases the heart rate and the stroke volume. At the same time, it causes the adrenal hormonal input by causing the adrenal medulla to release adrenaline, which increases androgenic receptors. Which increases the binding of adrenaline, which causes the heart rate to increase and contractility to increase again, and also thyroxine as well. So other things that can increase your heart rate is, caffeine. So that's why they say it's not very good to consume a lot of caffeine. And, caffeine is consumed within coffee and stuff. So too much of a coffee is not a very great idea because, it can cause your heart rate to increase. And if your heart rate, is increased above, flies out the roof, then that can cause detrimental effect on your blood pressure. Negative chronotropic factors, such as sedatives, usually given. Sedatives are basically painkillers or things that make you sleep. So those are, things that decrease your heart rate, and that can be also detrimental if it goes down significantly. If you administrate a really high dosage, they could potentially put the patient in coma or something. Now, age. Heart rate is also influenced by the age. Heart rate is fastest during the fetal stages, so it's beating around 140 to 160, and it usually gradually decreases through childhood and almost all of adult life, and the elderly commonly develop tachycardia. So tachycardia is very fast heart rate. So initially, when you're small and when you're a baby, your heart rate is usually faster than during your mid ages or during your childhood and adolescence, and then when you get to adult life, you develop tachycardia or sometimes bradycardia as well. Heart rate is faster in women, so gender is also an effect, whereas men has a heart rate of 64 to 72, and that's mainly because we have a bigger heart and a bigger muscle, heart myocardium, so there's a bigger surface muscle around the heart, so it doesn't need to beat as fast to push around the same amount of blood, right? And physical fitness, in terms of males, we're more physically active. I'm not saying everyone is, but males have a more muscular structure. So highly fit individuals have lower resting heart rate due to increased vagal tone and decreased sympathetic tone. Now the body temperature, so during hypothermia, which is when your body temperature is really high, as in fever or strenuous exercise, heart rate is increased. And this is because, when your temperature in your body really, exceeds a certain limit, um, you try to get rid of that heat, right? And the best way to do it is your blood arteries and veins, your capillaries near the surface of your hand and everything. You might learn this in biology during GCSEs or your A levels and stuff. They dilate so that the blood's is more exposed to the surface of the skin, allowing heat to be evaporated away from it. So hence the reason why the blood is pumped around more faster, so it's increasing heart rate. Now, decreased body temperature causes decrease in heart rate. Both conditions are associated with changes in metabolic rate as well as myocardium. Now, so far we've established things and factors that affect the heart rate. Right, now we need to establish the factors that affect stroke volume. So stroke volume is affected by something known as 1. Ventricular contractility. So the capacity of the ventricle to produce a certain amount of force. So the higher the ventricular contractility, the more cardiac output, because the more blood you're pushing around the body. The preload, and this is basically the amount of blood in the heart at the end. Well, to be really concise, there is a definition for preload, which we'll go into later. So just for now, you just need to know 3 factors affect stroke volume: ventricular contractility, preload, and you also have something called afterload. Okay, the preload is basically the same as end diastolic volume, and the afterload is basically the same as end systolic volume. So the first one, the influence of ventricular contractility. So any factor that causes an increase in contractility, as I said, is obviously going to increase stroke volume and vice versa. And this can be controlled by sympathetic nervous system or hormonal. So the sympathetic nervous system, stimulation of cardiac muscles, this is released by the we've already established that the neurotransmitter in this is norepinephrine or noradrenaline. Norepinephrine binds to beta-2 androgenic receptors on cardiac muscle cells on the membrane. This stimulates the second messenger cyclic AMP to open calcium 2 plus channels on the membrane. And if you remember back from the electrical activity video, or if you just recap on your notes from university or from your textbooks, you would know that during the conduction pathway in the electrical activities, calcium 2 plus that's involved in the plateau phase, which is phase 2 of the electrical activity of the cardiac muscle, when the calcium determines how powerful or the force of the contractions. The more calcium you have, the greater the contractions, therefore that increases stroke volume. Okay, so this is just the image taken from a book. So this just sums up what that was said on the previous slide. Sympathetic stimulation releases norepinephrine, and this initiates a cyclic AMP, so a cascade of reactions. So you can see on the cardiac muscle cell, where the mouse is pointing to, you have receptors on the cell known as beta-1 androgenic receptor. So the norepinephrine binds to this, and this is a G protein coupled receptor. This then causes activation of cyclic AMP via conversion of ATP to cyclic AMP. The cyclic AMP then activates a protein kinase, which inevitably, all you need to know is, increases influx, so it causes more calcium to get in, so therefore a longer plateau stage, so therefore a higher contractility and more force, so it contracts with more force basically. So we've established how the contractility, the influence of ventricular contractility on SV, and how the sympathetic nervous system controls that contractility. Now, how does the hormonal control? So you've established again before in the slides of heart rate, epinephrine, i.e. adrenaline, is circulating in the bloodstream. So this is adrenaline is a hormone that circulates in the bloodstream, released from the adrenal medulla. This binds to the beta-adrenergic receptors on cardiac muscle cells, and again they do the same process and stimulate second messengers such as cyclic AMP to open and calcium channels on the membrane. So again, increasing the force of contractility. So in summary, contractility refers to force of contraction at any given preload. So the preload meaning the amount of blood, or the preload is defined as, you can easily think of preload as end diastolic volume, so the amount of blood that is in the ventricles at the very late stages of diastole, just before systole. The greater the force, the more blood in the ventricles at the beginning of the systole, the greater the force of contraction, so therefore the more blood that's ejected from the ventricles. Okay, so therefore if there's a higher contractility and a higher stroke volume, you would expect the afterload, which is the end systolic volume, to be less. Now this is what we were going to go into detail. So Starling's law of the heart. Preload is the amount of tension or stretch on the ventricular myocardium, and the cardiac muscle fibers are stretched due to blood filling the chambers. The effect of stretching ventricular walls increases the force of ventricular contraction, and this is an example of intrinsic control of the heart. Okay, so all you need to know is preload is the amount of tension or stretch on the ventricular myocardium caused by the end diastolic volume. So if you have a more end diastolic volume, i.e. more blood remaining within the ventricles, so therefore they expand and push out on the walls of the ventricles, so therefore there's more amount of tension, i.e. there's a greater preload. And if there's a greater preload, it means there's a higher force of ventricular contraction. Now, the influence of preload on SV. Okay, so when the rate at which blood, Starling's law of the heart is, when the rate at which blood flows into the heart from the veins, so i.e. the venous return changes, the heart automatically adjusts to its output to match the inflow. So if you're doing a really extraneous exercise, your heart needs to accommodate and adapt to that environment. Otherwise, you'd be deprived of oxygenated blood to those respiring skeletal muscles and cells. Now, Starling's law is based on the observed changes that occur in EDV and preload as a result of venous return. Now, again, going back to the same thing. So if you have a higher EDV, i.e. a greater preload, it means greater force of contraction. So Starling's curve are shown in the next few slides. Within normalness, any factor that increases venous return will result in increase in preload and force of contraction. So this means that if you have an increased venous returns, which i.e. your heart is beating faster, so it's able to push deoxygenated blood round more quickly, so the heart is being filled back with deoxygenated blood. Therefore, the heart realizes it needs to beat faster so that there's a higher cardiac output, and it needs to contract with greater force, i.e. greater stroke volume, so it can push out more oxygenated blood. And that's what inevitably this slide is saying. Right, so initial curve is shown in the middle, and you can see the increased sympathetic activity. So it causes stroke volume to increase, and decreased sympathetic activity, meaning increased parasympathetic activity, would mean a decrease in the stroke volume. Now again, extrinsic factors that affect stroke volume, things like acidosis or increased extracellular K+, or calcium channel blockers as well. Now the afterload is the pressure the left ventricle must exceed before the aortic valve opens, right? And the afterload is basically, all you need to think of it is the end systolic volume, so the amount of blood that remains in the blood after ventricular contraction. Now this indicates how hard the cardiac muscle must work to push blood into the arterial system. Right, so an increased afterload means a decreased stroke volume and a decreased cardiac output. So mean arterial pressure, if that increases, right? So that means there's a higher afterload, which is not a great thing, because that would then mean you have a lower cardiac output and lower stroke volume. And a higher afterload also indicates you have a higher total peripheral resistance, so there's a greater resistance to push blood out into the aorta and the pulmonary trunk. Now, so far, I hope you've learned something from this video. So on the screen now is just a diagram, which is literally an overview of everything that we've gone so far. And so if you were to start from the top, so let's work our way from the top. So in the case of a crisis or an exercise or a physical or emotional trauma, you're in a shock or something, your body can take two direct ways. So first one is that we are going to concentrate on this one here. My mouse is following. So at times you increase the sympathetic nervous activity, right? The sympathetic nervous activity, which includes if you think back, it includes the neurotransmitter known as norepinephrine or noradrenaline. This causes an increase in heart rate, so therefore this causes an increase in cardiac output. Or similarly, again, if we go down the same branch, you're in a shock or something, the sympathetic nervous system increases. This can also increase contractility, right? So this increased contractility means increased stroke volume, because that means you have a higher ESV, so your stroke volume increases, hence your cardiac output again increases. Now, during exercise, there's another pathway, which is increased activity of muscular pump and respiratory pump. The muscular pump is located at the back of your calves. This then increases blood volume, and then this then inevitably increases venous return. Right? When venous return is increased, it means you have a greater preload, so therefore this then increases stroke volume, which increases cardiac output. Now, at times of low blood pressure or low blood volume, because you've suffered an hemorrhage or excessive sweating, what happens is increased renal activity, so your blood is conserved. When your blood is conserved, when your water is conserved, so less excretion via urine, the volume of blood becomes more diluted, and the volume of blood increases again, increasing the venous return. So therefore, increases the stroke volume, or the preload, which increases the stroke volume. Now, when in the case where when there's a very high blood pressure and very high blood volume, what happens is it reduces and inhibits, so it decreases signal to, and venous return decreases, and when venous return decreases, the afterload increases, so therefore stroke volume decreases, hence cardiac output decreases. Now, during times of high blood pressure, what you can have is you can have a higher ESV, so end systolic volume. If you have during high blood pressure, you have higher end systolic volume, which is bad because higher end systolic volume would mean you have a higher afterload, which decreases stroke volume. Okay, so hopefully you understand what I've tried to convey through this video. And if I haven't made too much sense, please do leave a comment below on how I can improve. Hopefully I have, if you do run back through the video carefully again, or you can also alternatively go onto our website at www.royalchristcollege.co.uk, which I would put in the description, and then go onto the cardiovascular section and find the cardiovascular physiology and download the PowerPoint. You can, alternatively go through yourself. It's always advisable to go through these slides along with your university lecture notes from your professors, and in combination with a set of textbooks like Tortora, Lippincott, or Medical Science, or whatever you have in whatever country you're studying. So on behalf of RCC, my name is Matthew Joes. It's been a pleasure making this video. Hopefully you like it, subscribe and share our video. And thanks again, once again. Thank you.